All right, let's talk about some biological molecules, or otherwise known as macromolecules. So, organic compounds, starting off, let's just define what does organic mean? Define organic. So, organic compounds are going to be compounds that contain both carbon and hydrogen. So, we have a few different types of macromolecules that we're going to be talking about. We've got proteins, carbohydrates, lipids, and nucleic acids. These might look familiar from your previous science courses, or maybe you've just seen on the nutrition facts of various products, proteins, carbohydrates, probably fatty acids, and then typically they omit nucleic acids as a macromolecule. So our proteins, those are going to be composed of multiple amino acids. So amino acids are going to be the building blocks for your proteins. When it comes to carbohydrates, you are going to be a chain of many monosaccharides. So monosaccharides are going to be the building blocks there, or monomers of sugars. Now, we'll just go to nucleic acids. So your nucleic acids are going to be composed of nucleotides. Now, lipids are going to be kind of unique, and we'll see that when we eventually get to it. They're going to have they're a little bit more nuanced, and we'll talk about those nuances later. So all of your macromolecules are going to be made up of a simple molecule called a monomer. So amino acids are a type of monomer. Monosaccharides are a type of monomer. Nucleotides, a type of monomer. And all of those are going to be linked together to make polymers. So your proteins are a polymer, a type of polymer. Your carbohydrates, a type of polymer. Nucleic acid, a type of polymer. And again, lipids are a little bit more nuanced. Before we deep dive into the macromolecules, let's talk about some functional groups that are going to be important moving forward in this class. So first, we'll start off with the hydroxyl group. So this is just going to be an oxygen bound to a hydrogen. So you might have noticed this from the last PowerPoint that I went over. Hydroxyl group, I mentioned hydroxyl groups. Those are going to be found typically not limited to carbohydrates and many other cellular compounds. So it's going to typically be found in these compounds, but it's not limited to these compounds. So you can find hydroxyl groups in many other compounds, just typically found in carbohydrates and many other cellular compounds. Next, we have our aldehydes. So these are going to be found in aldo-type sugars. One example of this is ribose. So this is going to consist of a carbon doubly bound to an oxygen and then also bound to a hydrogen. And then we have ketones. So found in keto type sugars, so things like ketose. Anytime you end in an ose, as a fun fact, things ending in ose are typically going to be some type of sugar. So this is going to be a carbon doubly bound to an oxygen. And then we have a carboxyl group. This is going to be a carbon doubly bound to an oxygen and also singly bound, so a single bond with an oxygen, which is also bound to a hydrogen. So typically, these are going to function as acids. This oxygen or this hydroxyl group will dissociate, and this hydrogen will be donated into something. So typically you're going to find these on organic acids, such as amino acids, as well as fatty acids. So what I've highlighted here in red is the hydroxyl group. Be sure you know that one out of all of these here. So next, we have the amine group. So this is going to be found in amino acids, and amino acids compose proteins, and all other nitrogen-containing compounds. Next, we have phosphates. So phosphates are very important. We're going to be talking a lot about phosphates in this class, and that's going to consist of an oxygen bound to a phosphorus, and the central phosphorus is bound to an oxygen, bound to an oxygen, bound to an oxygen. So it's just pretty much a phosphorus surrounded by oxygen. That's what a phosphate group is. And you're typically going to find this in DNA, RNA, and then also an ADP and ATP, adenosine diphosphate, meaning you have two phosphates, adenosine triphosphate, meaning you have three phosphates. And also you're going to find phosphates in your phospholipids. And then lastly, we have sulfhydryl groups. So these are going to be found in the amino acid cysteine. And thus, because cysteine is just a single building block, a single letter of the amino acid alphabet that could be found in a multitude of proteins, typically you're going to find sulfhydryl groups in most proteins. All right, and so this should definitely be review. Condensation or dehydration synthesis. So this is where we're moving from a monomer, so a single subunit, to a more complex polymer, a complex compound. And in the process of forming that larger, more complex molecule, we are removing water. So let's say we have a monomer here, and this monomer here, we want to combine these. We take the hydroxyl group from one and the hydrogen from the other, fuse those together, that's water, dehydration. We remove water, and what we do is we form a link between these two monomers. And then let's say we have this dimer here, and we want to fuse it with this monomer over here to make a trimer. Well, we remove hydroxide, the hydroxyl group, and we remove this hydrogen and dehydrate it, fusing them together, making a trimer or a polymer, you could say. And so typically in biological systems, these are going to be mediated by enzymes, and enzymes are typically going to be composed of proteins. So next we have the hydrolysis reaction. So I like to break down this word into two subunits, hydro meaning having to do with water, and lysis meaning to cut. So to cut something using water. So you're taking a complex polymer, a large macromolecule, and you're cutting it down into a simpler molecule, you could say, or simpler, a group of simpler molecules. So what you're doing is you're using water, you're adding water here, so H2O is going into the equation, and you're breaking apart these monomers from one another. So now we have a single monomer and a dimer over here. We started off with a trimer, now we have a monomer and a dimer. And that whole process was mediated by an enzyme, and that was a hydrolysis reaction. So we do the same thing here. You're adding water, you're using water, and so this hydroxyl group goes to this monomer, and the hydrogen moves to this monomer, and you're, in essence, breaking apart bonds by adding water to it. So that is a hydrolysis reaction. All right, so let's start off with carbs or carbohydrates. So we have three major functions of sugars. These aren't the only things that they can do, but for the most part, they're going to store their energy in their carbon-carbon bonds. So they have a lot of potential energy in their carbon-carbon bonds. Most sugars will. And those sugars can be transported, in your blood vessels to your tissues that are in need of sugars. And then they can also help compose carbon skeletons or cell walls, for example. So here we have cellulose. So cellulose, cell wall, and then you have a plasma membrane just underneath that. So this could be like a plant cell, for example. Okay. So your individual monomers are going to be called monosaccharides. They're the larger umbrella for your monomers are, or your sugar monomers are monosaccharides. So things like glucose, things like fructose. So glucose is a monomer, fructose is a monomer, and these are both isomers. What I mean by isomer is that they have the same chemical formula. So glucose here, fructose here, they have the same amount of atoms of every type. So they have the same chemical formula. However, the atoms are arranged differently. So we call them isomers. So glucose and fructose are just isomers of one another. Both of them are types of monosaccharides. Ribose is a pentose. That's a 5-carbon sugar. So these are actually 6-carbon sugars. Glucose is a 6-carbon sugar. Fructose is also a 6-carbon sugar. Next, we have disaccharides. So di meaning two, right? Di meaning two, mono meaning one. Disaccharides, that's just a bond between, or it is not just a bond between two sugars, but it is a compound that you create when you bind two sugars together. So the example that we have here is sucrose. If you bind glucose and fructose together, and if you look here, looks like you're removing this hydroxyl group and this hydrogen, dehydrating it, forming a bond between them. That would be dehydration synthesis, mediated by an enzyme, likely composed of a protein. You form sucrose, which is a disaccharide. Now, this bond between these two sugars is a glycolytic linkage. Very important to know. And then lastly, we've got oligosaccharides. Actually, this is not lastly, this is the third one. We have 4 to talk about. So oligosaccharides are going to consist of 3 to 20 monosaccharides. So these are going to be chains of sugars that will, sometimes they'll bind to proteins, sometimes they'll bind to lipids, and they can act as surface signals, much like what we see in, your antigens on the surface of blood cells that we associate with blood typing. And then lastly, we have polysaccharides. So these are going to be our complex carbohydrates. So things like starch and glycogen and cellulose. So starch, that's the way that your plants will store sugars. Glycogen is the way that animals will store sugars. And then cellulose, this is going to be an indigestible complex carbohydrate, typically found in plant cell walls. And we actually don't produce the enzyme to digest cellulose. So the thing is, it is still, much like starch, much like glycogen, a chain of monosaccharides. However, it's arranged in a way that is different from starch and glycogen, so we need a different enzyme to break them down, and we just don't happen to produce them ourselves. So because we can't break them down, we don't metabolize them for energy, so they just kind of run through us. They're a source of roughage, or you could say dietary fiber. Now, cows they're a great example of a mutualistic relationship between them and bacteria that live in their gut. The bacteria that live in their gut will actually produce an enzyme called cellulase. So ase, meaning it's an enzyme. Cellulase actually breaks down glucose. And so those bacteria in the gut of cows will break down cellulose into their subcomponents, and those subcomponents can then be broken down further by the cow's digestive system into energy, broken down into energy and those carbon-carbon bonds within those sugars. All right, so let's just look at, roughly what these things look like. So starch, typically you're not going to find much branching, but it's going to be water soluble. So it's soluble in water. So here's your starch, your cellulose. Usually your sugars are going to alternate in position. So if you could see structurally, it is different. It is water insoluble, meaning it's mostly nonpolar, because it's not dissolvable in water. Therefore, it's hydrophilic, right? And it's hypothesized to be the most abundant organic molecule on Earth, just because we have so many photosynthesizers on Earth. Okay, and then lastly, we have glycogen, which is going to be highly branched, and then it's also going to be water insoluble.